Okay, so hello everyone. So I think I just start the class for today and let others join afterwards. So there are some network issues from my end, so I hope that you can see the slides accordingly, but I will try to do my best to take the class properly. So please cope with it. I will just start the presentation right now then. Okay, so I hope that I am clear as well as I am audible. So welcome to the extra section for the week 9 section on the course, NPTEL course Cell Biology, Cell Organization, Division and Process. So my name is Isaac Cherian. I am a 5th year PMRF PhD scholar at School of Life Science, JNU New Delhi. So today what I will be doing this is that as this an extra section, like we will be discussing some of the like a few questions from all the week sections combined, like not for all the week sections, but from some of the important sections that we have covered till now from week 1 to week 8. So we will be discussing about like 30 questions, like all of them will be from my part that I made. So I think we can just start from the first question itself. So if you have any doubt in between, you can just unmute yourself, or you can ask, or otherwise you can type in the chat box. Okay. Okay. So coming to the first question. Isoforms of certain proteins in vertebrates prevent axons and dendrites from same neuron to make connections with each other. What are these proteins called as and are involved in prominently in vertebrates only? First option is DSCAM, second option is protocadherins, none of the above, both A and B. Which might be the answer? It is found in vertebrates, and it is something that isoforms of these proteins actually prevents the axons and dendrites from the same neuron to make connections with each other or interact with each other, because the neurons should interact with other neurons, right? Dendrons of nerve endings of one neuron should interact with the dendrites of the other neurons. What may be the answer? Anyone? Anyone who know the answer, might be? Okay, so the answer to this question is protocadherins. So basically option A and B might be the answer, but D scam, the first option one, right? They are found predominantly in case of Drosophila. So in case of vertebrates, from the question itself, it is said that it is predominantly found in vertebrates, right? So the answer to this question is protocadherins. D scam also has the similar function in case of Drosophila. What it does is that it has several isoforms itself. It can undergo different forms of it can different isoforms by a process called as what is it? Alternative splicing. While the it can form different isoforms. So identical, some of the nerves that are present in neurons that are present in Drosophila, right? If consider that there are two neurons, so both of them have the same isoform of DSCAM. So they can dictate itself to each other, and because of the same isoform, it provides kind of a repulsive nature in this case. So the nerve endings of dendrites cannot interact with each other. So basically, because of the presence of this isoform, right? So in case of say neurons, the dendrites, in case of same neuron, the same isoform will be present there. In the nerve endings and the dendrites, same isoform of DSCAM will be present there. So they cannot interact with each other because they are repulsive to each other. But the different neuron, different isoforms will be there. Because of the presence of different isoforms of DSCAM, what it does is that they can interact with each other because there are no repulsion, because they are not identical in nature between different neurons. So DSCAM is one such protein that is found in Drosophila, while protogathering is found in vertebrates, and it also has a similar function as to what is found in case of DSCAM. So both are present there, but and in case of vertebrates, DSCAM is also present there, but it is not prominent. The functionality is less prominent as compared to what is seen in protogatherings there. So that's the function. It is commonly found in Drosophila, fruit flies, where it also mediates self-avoidance. However, DSCAM is also present in vertebrates, but its role in vertebrates is less prominent compared to protogatherings in terms of self-avoidance. These are part of gathering superfamily. From the name itself, you can see that it's a superfamily of gatherings, belong to a gathering superfamily, and contribute to self-awareness through their different isoforms formed by different processes like alternative splicing and all, and which can mediate cell-cell interactions and repulsion between dendrites of same neurons. I hope this answer is clear. So anyone has any doubt? Okay, 1 minute. I think I am having some network issue. I will just sort it out. Okay, sorry for that. I am having some network issue here. So anyone has any doubt? Okay, so I think it's quite clear. So I will just move on to the next question then. Okay, coming on to the next question. Okay, which of these proteins have a dephosphorylation activity? CA kinase, cyclin activating kinase, V1, CDC25, or none of the above? Which may be the answer. Out of these proteins, which of them have a dephosphorylation activity, in that it can remove proteins from certain proteins? In this case, say like certain CDKs and everything. I am getting option number 2. Any other option? Anyone else has any other answer? Okay, so the answer to this question is option number 3, CDC25. CAK or cyclin activating kinase and V1 kinase, they are both kinase, meaning that it has a phosphorylating activity, while CDC25 is a dephosphorylase. From this you can see that this is CDC2, right? So basically, in order to initiate the meiosis phase, CDC2 is important. See, it binds to cyclin B and O. So in case of CDC2, which is a cyclin dependent kinase, it consists of 3 important residues, that is threonine 161, tyrosine 15, and threonine 40. What CAK, that is cyclin activating kinase, and V1 kinase does is that it phosphorylates all these 3 important proteins. So threonine 161 is called as a activating amino acid ratio, activating amino acid ratio in case of CDC2 and all, while tyrosine 15 and threonine 14 are called as inactivating amino acid ratios that are present in CDT2. So whenever these 2 amino acids are getting phosphorylated, the CDC2 that is bound to cyclin is inactive in case it cannot show any kinase activity or phosphorylating activity. What CDC25 does is that it is a dephosphorylation activity that you have discussed before, right? So what it does is that it removes the phosphates that are present in both tyrosine 15 and threonine 14, rendering it free dephosphorylated, while the threonine 161 that has been phosphorylated earlier remains phosphorylated there. As it is an activity in amino acid residues, and only that particular amino acid residue is phosphorylated, right? So what happens is that this CDT2 with cyclin B is getting activated there, because only the threonine 161 is getting phosphorylated there. Whenever it is getting activated, it can like perform several functions that are required for carrying out the early mitotic stage and all with cyclin B. So whenever it gets dephosphorylated, and when the anaphase promoting complex are getting activated, cyclin B gets degraded, and then the mitosis can enter into like anaphase, and then can enter into another stage of interphase, that is the G1 phase. So CDC25 is the phosphorylating dephosphorylating phosphatase there, whilst B1 and CAK are both kinases. So I hope I am clear. So anyone has any doubt? Anyone has any doubt regarding this? These all sections like we have discussed from I think week 2 or week 3, right? So I think this is an important section there, so I think you should go through it. If anyone has any doubt, you can unmute yourself and then ask. Okay, so I think I am clear. So just go through each section, right? So I think it's an important section. So I will just move on to the next question. So coming on to the next question. Out of this neurotransmitter, which one is involved in regulating mood and appetite? Acetylcholine, dopamine, serotonin, or glutamine? Which of this neurotransmitter is involved in regulating mood and appetite? Anyone? Yeah, I'm guessing option number 3, serotonin. Anyone else? Does anyone else has any other answer? Okay, I think majority of you go with option number 3, right? So option number 3, serotonin is the right answer. So basically serotonin is out of this neurotransmitter is involved in regulating mood and appetite. It is associated with other functions like regulating your emotional well-being, like maintaining your calmness and happiness, and also plays an important role in regulating your appetite suppression as well as digestion. Other components like acetylcholine, it is an important neurotransmitter that is present in like neuromuscular junctions, right? Autonomic nervous system and brain. So autonomic nervous system controls the involuntary system of the body, and it also has an excitatory effects on muscles, especially in case of skeletal muscles, smooth muscles, etc. This acetylcholine, when is released at that particular junction, it can carry out several excitatory function. While in case of other functions, like in case of regulating the cardiac muscles that are present in heart and all, it can be either be excitatory or inhibitory nature. Other functions are also there, but at this point I think this much is important. And also in case of dopamine, it is released in brain as well as that is in central nervous system as well as in peripheral nervous system, and it has both excitatory and inhibitory function. It also regulates the emotional state of or well-being of that particular individual. While in case of glutamate, it is present in case of this brain as well as spinal cord, that is peripheral nervous system, and it is mainly in case of central nervous system, that is the brain, it is excitatory in nature. So all of these neurotransmitters are important, but sometimes most of the neurotransmitters like acetylcholine are usually excitatory in nature, especially in case of muscle activity and contraction everything. While the others are important, but most of them are inhibitory, can be either be inhibitory or excitatory in nature. So there's a difference when you are when some questions like this is getting asked and all, so you must like keep in mind about these points and everything. So I hope it is clear. So anyone has any doubt? Okay, so I think my part is clear. So I will just move on to the next question. If you want me to repeat something, like I can repeat it, no issue. You can just type in the chat box, right? Okay, so I will just move on to the next question. So coming on to the next question, what is the function of tau protein in DNA replication? It is something that is associated with DNA polymerase. So it helps to load sliding clamp onto a primer template duplex. It prevents the annealing of DNA duplex. It helps to connect the clamp loader to DNA polymerase. It helps to synthesize RNA primer to initiate DNA replication. What is the function of tau protein? Out of these options, which might be the answer? Yeah, I'm getting option number 3. Anyone else? Okay, so you are basically right. Option number 3 is the correct answer. It helps to connect the clamp loader to DNA polymerase. So this is the basic structure of that DNA polymerase along with the other components that are present in that particular complex. You can see that there is a sliding clamp, there is a sliding clamp loader, tau proteins, as well as 3 core DNA polymerases, right? So what happens is that whenever a DNA is getting unwound into a single stranded molecule by a helicase, right, these clamp loader proteins bind to specific regions that are present in the DNA at that near to that helicase, and whenever this sliding clamp is bound to it, the sliding clamp loader along with the tau protein as well as 3 core DNA polymerases are bound to it. So there are 3 core DNA polymerases because one core polymerase is responsible for the replication of leading strand, while the other 2 DNA core polymerase are sequentially exchanged and exchange with each other and helps in the synthesis of this lagging strand outdoor. So this sliding clamp insert itself into the annealed DNA sequence, and the clamp loader also helps in that particular function. And whenever this clamp loader is bound to us, this tau protein is getting attached to it, and the tau protein acts as a linker protein that connects the polymerase 3 to the sliding clamp loader there. So it is, as you can from the structure, it is quite long and it is quite thin, as well as it is flexible in nature. Because of the flexibility of this tau protein, the DNA polymerase can actually help in efficient synthesis of the both lagging as well as the leading strand in this case. So and it is quite flexible and can move at different angles. So whenever a DNA polymerase is sliding through the annealed DNA strand, right, double stranded DNA, it can actually needs this tau protein because of the flexibility that is provided. So it can like synthesize those particular strands wherever it is needed. So that's the function of the tau proteins, and it helps to load the sliding clamp onto a DNA template duplex. The option 1, that is the function of the sliding clamp loader, and also it prevents annealing of DNA duplex. That's the function of like there are certain proteins that prevents the annealing of the DNA duplex, right? It maintains it in a single stranded fashion. So what proteins might be involved in that? It prevents the re-annealing of the DNA duplex. Which proteins actually carry out that particular function? Anyone? Okay, though, so the specific function is carried out by what is called as a single stranded binding proteins, right? SSB proteins. So those binding proteins actually bound to specific regions in the DNA, preventing it from annealing it to a DNA duplex. It remains in the single stranded DNA. Only then DNA replication can take place. And it helps to synthesize RNA primers to initiate DNA replication. So this is carried out by a protein called as primase, right? So primase helps in the synthesis of RNA primers. RNA primers are involved in DNA, initiating DNA replication, not DNA primer. So afterwards, whenever the DNA strand is synthesized, the RNA primers are removed and then it is replaced by DNA nucleotides at that region. So RNA primers initiate the DNA replication and it is produced by primase enzyme, right? So in this question, the top protein helps to connect clamp loader to a DNA polymerase. So anyone has any doubt? Okay, so I think I'm clear. So I will just move on to the next question. Okay, coming on to the next question. CDK1 and cyclin A helps in regulating which phase of the cell cycle? G2 phase, G1 phase, S phase or M phase? Cyclin A and CDK1. Cyclin B and CDK1 are involved in mitosis. The cyclin A interacts with CDK1 at which phase of the cell cycle? Yeah, I'm getting option number 1. Any other option? Anyone else has any other answer? Okay, so the answer to this question is you are right. Option number 1, G2 phase. So this is the structure of the whole cell division phase, right? So you can see that if you start with G1 phase, cyclin D along with the CDK4 or CDK6 is essential for regulating its function. So in the early phase, cyclin D binds with CDK4, while in the later phase, cyclin D binds with CDK6. However, in order to transition from G1 to S phase, cyclin E is essential, and for that, cyclin E incorporates with this kinase, that is CDK2, and helps in the transition. So there is a phase called as S phase there, right? Start phase there. Start is actually a checkpoint. So in order to pass that, cyclin E should interact with CDK2. So whenever it is passed, it interacts into S phase. So throughout the S phase, cyclin A interacts with CDK2. So you have to differentiate the distinction there. In CDK, it can also interact with the CDK1 as well as CDK2. But in case of S phase, cyclin A interacts with CDK2. So it regulates, it is present at all phase of cell cycle. However, when the transition needs to happen, either cyclin B or cyclin A need to interact with CDK1 so that it can interact into G2 phase and further move on to M phase. So cyclin B or cyclin A interacts with this particular CDK1 so that the transition can happen from S phase to G2 phase, and it is found throughout the cell cycle. However, when G2 phase need to be transitioned into M phase, this cyclin B interacts with CDK1, and then this transition into M phase takes place because of this particular interaction of cyclin B with CDK1. And then to early phase anaphase promoting complex become active, it degrades out the cyclin B so that anaphase can happen, and further on it can, once telophase and cytokinesis happen, M phase can transition to G2 phase. So these CDK cyclin interactions are important. I think you should memorize that also. So yeah, so which cyclin interacts with CDK, which the number differentiate, right? It's different in this case. So you should memorize that in this case. So I hope I am clear. So anyone has any doubt in this case? Okay, so I think it's clear, right? So as there are no doubts, I will just move on to the next question. Coming on to the next question. Spindle assembly checkpoint is seen in which phase of the cell cycle? S phase, G1 phase, M phase or G2 phase? Spindle assembly checkpoint. This is an easy question. Option number 3 I am getting. Anyone else? Okay, so basically you are right. So answer to this question is M phase. So several checkpoints are present in different phase of cell cycle, right? So in majority of the case, like it is present. So one of the common checkpoints is like most important checkpoint is spindle assembly checkpoint. So in case of what this checkpoint does is that whenever a metaphase to anaphase transition needs to take place, right? Whatever happens is that to the kinetochore of the chromosome, the spindle fiber need to get attached. So only then, whenever to the sister chromatid, the spindle fibers to opposite kinetochores are getting attached, only then the chromosomes can be pulled apart and sister chromatids can be separated to each other, and then only a metaphase to anaphase transition take place, right? It is completed at that stage when they are separation, the sister chromatids are separated from each other. So this spindle assembly checkpoint, what it does is that it checks whether this particular interaction has taken place, this particular interaction of kinetochore with spindle microtubules has taken place. Only then this particular checkpoint is getting passed there. Otherwise, what happens is that the cells will get arrested in this stage if it knows that, or whether it has passed through a stage, the cell will not grow afterwards, or it will undergo apoptosis and the cell dies out. So this particular spindle assembly checkpoint is present in M phase, and it is essential for transition from metaphase to anaphase stage and subsequently into G1 phase and everything. Otherwise, it will get arrested in this particular M phase itself, that is in the metaphase stage. So other checkpoints are also present. So the in both all the three cases, in the G1 and S phase as well as G2 phase, DNA damage checkpoints are present there, right? So there are several like checkpoints like ATR as well as ATM checkpoints that actually regulate these particular checkpoints. What it does is that it checks the DNA that is formed within the synthesis phase of the S phase, right? It is also called synthesis phase, whether the DNA is getting damaged or not. If the DNA is getting damaged, or because any double stranded or single stranded break is there, what happens is that the cell will get arrested at this particular checkpoint. If it has the S phase, then it will get arrested at the S phase. If it has the G2 checkpoint, then it will get arrested at the G2 checkpoint accordingly, or the G1 phase, it will get arrested in the G1 phase. So in all the three cases, DNA damage checkpoint is present, but in case of M phase only, spindle assembly checkpoint is there. So yeah, so the basic answer is there. So different checkpoints are present that actually regulate the transition from one phase to other, and these are important in case of cell cycle or cell division and everything. So I hope I am clear, and specific spindle assembly checkpoint is present in M phase. So is there any doubt regarding this? Does anyone has any doubt? Okay, so I think it's quite clear, so I will just move on to the next question. So coming on to the next question, the Nyanmar motif in the origin of replication complex, that is ORIC, is involved in which of these functions? Binding to initiate a protein DNA, site for the formation of single stranded DNA, binding site for the helicase, or site for the generation of RNA primase. Which might be the answer? There are two motifs that are present in RIC. We will discuss that. One of them is Nyanmar, out of which these options, which might be the particular activity that it might be carrying out. Okay, I am getting option number 3. Anyone else has any other option? Okay, so the answer to this question is binding to initiator protein, that is DNA A. So that is the first option there, right? So basically this is the structure of in case of Escherichia coli, this is the structure of the region that is present in DNA, that is ORIC, origin of replication complex. So it is about 245 base pairs in length, and there are two different motifs, that is 13-mer motif and a 9-mer motif. And if you consider the 13-mer motif, there are 3 such motifs that are present here. You can see that there are 3 red boxes, right, and they denote as 13. So there are 13 nucleotides here, and they are quite similar to each other, and it is called as a 13-mer motifs, and there are 3 such motifs that are present there. While in case of 9-mer motif, you can see that they are present at different regions in different length. In case of 13-mer, they are quite close to each other, while in case of 9-mer, they are quite far away from each other. There are 5 such motifs that are present there. You can see that 1, 2, 3, 4, 5 such 9-mer motifs are present there, right? So what it does is that the 9-mer motif actually binds to initiator protein DNA. In case of E. coli and all, there are DNA G, etc., right? So each and every one has their function. One is a helicase, one is a primase, etc. And DNA A is the initiator protein that is present in E. coli. So this 9-mer motif has this DNA A, that is the initiator protein, has a tendency to bind to this 9-mer motif that is present in that is origin of replication complex there. While the 13-mer has a function of is the region where single stranded DNA is formed there. So these have essentially have this adenine thymine rich region and all, right? So they have these 2 hydrogen bonds that are present there. And while guanine cytosine region has a 3 hydrogen bond there, right? So adenine thymine region, because of the presence of only 2 hydrogen bonds, they are easily getting cleared out because of the number of bonds, difference in the number of bonds that are present between guanine cytosine as well as adenine thymine. So this single stranded DNA formation actually happens at this 13-mer region, while the DNA A actually binds to this 9-mer region. So that's the basic difference between those two. While coming to a third option, that is binding site of helicase, that actually happens within the ORIC complex, right? Like origin of replication complex is getting bind to specific region of the DNA, and helicase bind to both the sites of the ORIC complex, origin of replication complex there, right? You might have read that. And site for generation of RNA, that is RNA primase, that is carried out by the primase enzyme and all, and that also happens once the DNA is getting annealed, and to specific site the RNA primase are getting formed to initiate the replication there. So this is the function the 9-mer motif in ORIC is involved in, that is here, is binding to the initiator protein, that is DNA, to initiate the replication. So I hope it is clear. So anyone has any doubt? Okay, so I think it's clear, so I will just move on to the next question. Coming on to the next question. This is an easy question, basic question one. So what is the average length of Okazaki fragments? 10 to 100 nucleotides, 100 to 2000 nucleotides, 4000 to 6000, or 6000 to 8000 nucleotides? Which may be the answer? The average length of Okazaki fragments. Okay, so I am getting the answer 1000 to 2000 nucleotides, 100 to 2000 nucleotides. I think you mentioned the option number B, right? Okay, so basically you are right. So it exists between that range. Basically 1000 to 2000 is also right. But if you are considering it a broader range, 100 to 2000 is the specific answer. So you are right. So option number 2 is the correct answer. So basically these are the Okazaki fragments, right? So you can see that as we have discussed before, the DNA polymerase in case of vertebrates and all, it consists of 3 DNA core polymerase. One synthesizes the leading strand, other two sequentially helps in the synthesis of lagging strand. So you can see that the DNA replication actually happens in 5' to 3' end, and the DNA polymerase moves in this direction as well. So in this lagging strand also, DNA polymerase needs to move in this direction. But in case of us, you can see in case of lagging strand, it needs to be if it is moving in the particular direction and DNA replication is taking place accordingly based on the what happening in the leading strand, it should be synthesized in 3' to 5' end. But that is quite inefficient and energy consuming, so that cannot happen. So in case of lagging strand, DNA polymerase moves through its end and DNA strand is synthesized accordingly in 5' to 3' end. But what happens in case of lagging strand is that the lagging strand actually loops around the DNA polymerase, so that the direction is getting shifted there. So what happens is that because of this looping of DNA around the DNA polymerase, and so as the DNA needs to be synthesized in 5' to 3' end, so the DNA that is formed at that particular end is actually discontinuous in nature, and this discontinuous DNA is actually meshes between the length of 100 to 2000 nucleotides. So this particular fragments were first discovered by the scientist Okazaki, and based on his name itself, these were named as Okazaki fragments. These lagging strands that are being generated here are called as Okazaki fragments. And because of the looping over of this lagging strand around the DNA, so that the DNA can be efficiently synthesized in the 5' to 3' end, discontinuous stretches of DNA is formed there, and this DNA is also called as Okazaki fragments and is usually of length 100 to 2000 nucleotides. So these are called as Okazaki fragments. So I hope I am clear. So does anyone have any doubt regarding this? Okay, so I hope it's clear. I will just move on to the next question if there is no doubt. Okay, coming on to the next question. Growth of daughter centrioles are completed at which phase of the cell cycle? G2 phase, S phase, M phase, or G1 phase? Which might be the answer? Growth of daughter centrioles are completed at which phase of the cell cycle? Yeah, I'm getting option number 3. Any other option? Anyone else? Okay, so the answer to this question is G2 phase, because M phase cannot be answered, right? Because like M phase, the centrioles need to be prepared there, right? Centrioles are actually part of a centrosome there. So in centrosome there are 2 centrioles, and each of the centrioles are essential for maintaining the polarization of microtubule and helps in its assembly, microtubular assembly, which leads to a formation of spindle fibers there, right? So that's the basic function of centrioles. Sorry, centrosome. So centrioles that are present in centrosomes actually carry out this function. So what happens is that before the M phase starts, it needs to be present there, right? So it needs the growth of the daughter centriole should be completed before the M phase is getting released there. So in case of G1 phase, you can see that the cell is getting divided. So you can see that this is the centrosome. I hope you can see. this is one centrosome here, this is one centrosome here. When the DNA gets duplicated, one of the daughter will get one centrosome from the parent DNA cell, while the other will get the other centrosome there, right? So in case of when the S phase start, what happens is that this centrosome starts to get duplicated from each other. You can see that each of the centriole is getting divided. One is the parent one is the daughter one there, right? The orange one, it is the daughter one, while the green one, it is the parent one. So each of the centrioles that are present there start to get divided in case of S phase. And this growth is completed, this division is getting completed, or the growth of the centrioles is actually getting completed in the late S phase or the G2 phase. You can see that with a single centrosome, but there are two centrioles that are present there. So afterwards, but the centrioles are actually, the growth of the centriole is completed at this phase. When the early prophase is getting reached, what happens is that these two centrioles are divided from each other, and there is one centrosome consist of two pair of centrioles there, right? So this centrosome division happens in the early prophase. So the centrosome get divided. There are two centrosomes there, having only one pair of centrioles there. So afterwards, whenever the cell division is completed, the cell continues. The cell, one of the cell gets one of the centrioles there, and the other cell gets the other pair of centrioles. Sorry, other centrosomes there, consisting of two centrioles that are present in that case. So growth of the daughter centrioles are completed at the G2 phase of the cell cycle, and it should be completed before the M phase, so that the spindle assembly can take place, so that the homologous chromosome can get separated each other, and histochromatids are getting separated each other, and hence telophase can happen, followed by cytokinesis, so that the two daughter cells are formed in the G1 phase. So that's the answer basically. In the G2 phase, the growth of the daughter centrioles are completed. So anyone has any doubt regarding this? Okay. So I think it's clear, so I will just move on to the next question then, right? Coming on to the next question. In peripheral nervous system, which of the glial cell is responsible for the formation of myelin sheath? Astrocytes, Schwann cells, microglia, or oligodendrocyte? Which may be the answer. In the peripheral nervous system, which of these glial cells are responsible for the formation of myelin sheath? Yes, I am getting option number 2. Anyone else? Okay, so basically, sorry, I had to, in the peripheral nervous system, right? In the peripheral nervous system, Schwann cells are the basic answer. So I will just correct it out. This is a mistake. So in case of central nervous system, oligodendrocytes are the basic glial cell that are responsible for the formation of myelin sheath, right? So basically, Schwann cell, what it does is that these are kind of flexible cells. So what it does is that it binds to the axons that are present in the nerve fibers there. Consider this is in the peripheral nerve system. Consider that this is the axon. So there are two types of neurons there, right? Myelinating and Schwann cells there, right? Myelinating and non-myelinating Schwann cells. Non-myelinating ones are just involved in maintaining the structural and integrity of the axons there, while in case of myelinating Schwann cells, they help in the formation of myelin sheath there. So Schwann cells cover over the axons at particular region called as internodes that are present in axons, and forms the what is called as myelin sheath there. So the region that is present between two internodes that are absent of this myelin sheath is called as nodes there, or nodes of Renvier there. This is helpful in maintaining the conduction that is happening for single transition along the axons and all. So this is happening in case of peripheral nerve system. Sorry for this, I will just change this option there. It's Schwann cells basically. So what oligodendrocyte is there does is that in case of central nervous system, it carries out the function of Schwann cells there. In case of central nervous system, this you can see that there's microglia, astrocytes as well as oligodendrocyte, right? So oligodendrocyte acts as Schwann cells in case of central nervous system and helps in the formation of myelin sheath across the axons of the neurons that are present in central nervous system. While microglia is also called as the brain's immune system, it helps in providing immunity in case of central nervous system and all, while astrocytes helps in maintaining the osmotic balance and also providing the structural as well as functional aspect of regulating the functional aspect of neurons that are present in the central nervous system. So these are called as glial cells, that is microglia, astrocytes, and as well as dendrocyte in case of central nervous system, while in case of peripheral nervous system, Schwann cell is a glial cells that are present there. So in case of peripheral nervous system and central nervous system, there is a distinction or difference between the cells, glial cells that are present there, or whatever function it carries out. So I hope this is clear. In case of peripheral nervous system, Schwann cells are the one glial cells that is responsible for the formation of myelin sheath there. So anyone has any doubt regarding this? Okay, so I think I am clear. So I will just move on to the next question then. Sorry. Okay, coming on to the next question. Middle lamellae of plant cell is composed of which of these substances? Cellulose, pectin, hemicellulose, or fatty acids? Middle lamellae in plant cells are composed of which of these components there? There are different components that are types of cell walls that are present there. The primary is there, secondary cell wall is there, as well as middle lamellae. Each of these have different constituents that are present between them. So in case of middle lamellae, which of these options are present? Yeah, so the answer to this question is option number 2, pectin. So basically, like you can see this structure, right? So there is a plasma membrane there, followed by the plasma membrane there is a primary cell wall, middle lamellae, and secondary cell wall is also present in some cases and all. Majority of the time these two are present there. So you can see that in case of primary cell wall, the cellulose is present there, and along with this hemicellulose is present there. So cellulose and hemicellulose actually exist as polymer there, right? So that's why it's called as cellulose microfibril there. And you can see that there are also hemicellulose that is present there. Hemicellulose is usually branched in nature, and other proteins, soluble proteins, are also present there. That doesn't matter. While in case of middle lamellae, you can see that majority of the pectin, some amount of pectin is also present in primary cell wall, but majority of the pectins are occupied within this middle lamellae and everything. So what pectin is that it actually combines with several divalent cations like magnesium and calcium to form magnesium and calcium pectate, which overall maintain the structural integrity of the cell wall. So together with primary cell wall, this middle lamellae and primary cell wall helps in maintaining the structural as well as other functional aspect of the plant cell walls there. So in case of plasmodesmata, you can see that the outer layer is made up of middle lamellae followed by primary cell wall there, right? So those are basically some of the functions that middle lamellae carries out in this case. And whenever the cell plate formation is taking place in case of plant cell, middle lamellae is the one that actually forms first whenever the cell plate is getting formed, followed by cytokinesis and all. So I hope I am clear. So anyone has any doubt regarding this? Okay, so I think it's clear. I will just move on to the next question. So coming on to the next question. What is the role of P21 in cell cycle checkpoint? It regulates the assembly of spindle fibers. It inhibits the formation of CDK2 cyclin E or A complex. It activates P53 that enables DNA repair. It prevents the transition from M1 to M phase to G1 phase. Which might be the answer? What is the role of P21 in cell cycle checkpoint regulation? Okay, I am getting option number 4. Any other answer? Anyone else? Okay, so the answer to this question is option number 2. It inhibits the formation of CDK2 cyclin E or A complex. So this is the basic function. You know that the P53 there, right? So what it happens is that P53 is actually kind of also called as a tumor suppressor gene there, right? Whenever it is active, it suppresses the like tumor generation, and it promotes apoptosis at certain cases and all. So what it does is that it acts as a transcription factor and binds to this P21 region, that is bind to the promoter region of P21 and helps in the generation of this P21 there. So P21, what it does is that it binds to the cyclin CDK2 cyclin E or A complex, and what is this, it acts as a regulatory mechanism for regulating the start checkpoint that enables the transition of G1 to S phase there, right? So start checkpoint is present with at the end of the late G1 phase there, right? It helps in transition to S phase. So whenever this P21 is active, what happens is that this P53 actually recognizes some kind of DNA damage that is present, that is happening there. Whenever it is present there, P21 is getting transcribed by P53, which acts as a transcription factor there. This P21 actually binds to the cyclin EA or CDK2 complex, and furthermore, because of this binding, what it does is that it inhibits the cyclin CDK2 phosphorylate this RB protein there. So whenever RB protein is not phosphorylated, it is always bound to this ETF, which is a transcription factor there. So whenever ETF is bound to RB protein, it cannot bind to the DNA promoter regions, that leads to the formation of several S phase cyclin genes and everything. So whenever S phase cyclin genes are not synthesized there, the cell cycle arrest actually happens within the start checkpoint there. So this is how start checkpoint is getting regulated by this P53 as well as P21 along with the RB E2F transcription factor complex there. So RB remains attached to E2F, so cyclin phase genes are not synthesized there, S phase genes are not synthesized there. So the transition from G1 to S phase like complex cannot take place. So that's how this cell cycle checkpoint or start phase checkpoint actually works out. So it regulates the assembly of spindle fiber. That's not the function of P21. It activates P53. No, P53 actually activates P21. That's the totally the opposite one in this case. And it prevents the transition from M1 to M to G1 phase. No, that's actually the regulation of like spindle fiber spindle assembly checkpoint there, right? P21 has nothing to do with spindle assembly checkpoint. So that's a totally dependent another checkpoint that is present between at the end of the M phase, which helps in M1 to M2 G1 phase transition. So it inhibits the P21, inhibits the formation of CDK, cyclin EA complex there because of binding to it. So I hope I am clear. So anyone has any doubt? Okay, so I think it's clear. So I will just move on to the next question, right? Okay. So coming on to the next question. Out of which of these proteins, which one of these is palmitoylated? Palmitoylation is actually a addition of a fatty acid to a particular either the N terminal or a C terminal of the protein depending on the protein. So out of these proteins, which of these show palmitoylation? ERK, MEK, RAS, or none of the above? Which might be the answer? There is a specific pathway that you have learned in the lecture video. If you have seen that pathway, then you will know the answer. Out of which of this protein is palmitoylated? Okay, so the answer to this question is RAS. So basically you have seen this pathway, right, receptor tyrosine kinase, which helps in the synthesis of this cyclin D gene, which is essential for this G1 phase maintenance and everything. So basically what is RAS is a protein is that it can exist in two different forms. It can be palmitoylated or unpalmitoylated. Whenever it is palmitoylated, it can either go into a ER membrane, or whenever it is palmitoylated, or can also act as another transcription factor whenever it is bound to GTP form. Whenever it is palmitoylated, it actually binds to a plasma membrane because it is a lipid chain, right? Palmitoyl group, palmitoyl-CoA you might have heard. So it helps to bind to the plasma membrane. So it assists at that particular range. So whenever any growth factor or anything is coming out, this receptor tyrosine kinase is getting phosphorylated. It undergoes autophosphorylation. So whenever it is getting undergoing autophosphorylation, other proteins are getting bind to it, like GRB2 as well as those proteins, and this enables the transfer of GTP into a RAS group, RAS protein there, which is palmitoylated. Whenever GTP is bound to RAS, this palmitoylation is getting removed there, and it assists as a soluble protein, which is in the form of RAS GTP. This in turn activates some downstream pathways like RAF, MEK kinase or ME kinase or ER kinase also, which then goes into a nucleus and activates the binds to a promoter acting as a transcription factor for cyclin D, and also helps in the synthesis of this cyclin D and other factors like MYC. So this helps in the crossing of the cell cycle checkpoint there, which enables the transition phase of cell division. So this is the basic functionality of RAS protein there. It is actually a part of this receptor tyrosine kinase protein that helps in the synthesis of the cyclin D genes and everything. So yeah, so I hope I am clear. So anyone has any doubt regarding this? And from the previous diagram you can see that cyclin D is actually involved in this G1 phase regulation and everything. Anyone has any doubt? Okay, so I think it's quite clear, right? So I will just move on to the next question. Okay. Okay, coming on to the next question. During mating type switching in case of yeast, which of these locus is cut by HO endonuclease? HML locus, homothallic mating left locus, homothallic mating right, or HMR locus, MAT locus, or both HMR as well as MAT locus? Which might be the answer? Okay, so the answer to this question is MAT locus, right? This we have discussed in the last week. So basically you can say that mating switching takes place in chromosome 3, right, based on the regulatory activities that is happening here. So this is the homothallic mating left locus there, and after the centromere you can see that MAT locus as well as HMR locus is found there. So there is a HO endonuclease site that is present in the MAT alpha locus there. So whenever mating switching need to take place, there is a looping of this particular chromosome so that the HML locus comes close to this MAT alpha locus there. So afterwards, whenever this happens, the HO endonuclease that is present there actually cuts this particular site there. So it leads to a formation of a double stranded break there. So this particular break needs to be actually what you can say repaired out there in this case. And what happens afterwards is that because of the homologous regions that are present between HML as well as MAT locus there, homologous recombination take place. So afterwards the DNA double strand repair take place. But because in case of homologous recombination that is taking place, this particular region that is found there, this particular region that is found there is used as a template for actually regenerating this particular region that is cut out by this endonuclease there. So you can see that the locus here becomes similar to that of MAT locus. The region the MAT locus becomes similar to that is found in the HML locus there, right? So whenever this transition takes place, the mating type switching of yeast happens basically. So the G1 phase gets arrested there, so that the two yeast strands that are present there, right? So they can actually undergo fission and leads to a formation of a diploid spores, diploid cell whenever they are in a haploid nature in this case. So I hope I am clear. This actually leads to a formation of mating type switching. This is because of the presence of release of several pheromones from a particular strain of yeast that is getting bound to certain receptors, G protein receptors that are found at the other similar identical type of yeast strain there, which actually promotes this mating switching and everything. So I hope I am clear. Does anyone have any doubt? Okay, so I think it's clear, so I will just move on to the next question. Okay, coming on to the next question. Hydroxyurea is an inhibitor of which of the following enzyme? Ribonucleotide reductase, HMG coenzyme reductase, carboxylic acid reductase, or thioredoxin reductase? Hydroxyurea. Okay, so the answer to this question is ribonucleotide reductase. So basically what this hydroxyurea does is that it binds to this ribonucleotide reductase and inhibits its function. So what ribonucleotide reductase does is that it helps in the conversion of this ribonucleotide into deoxyribonucleotide. So these ribonucleotides are essential for the synthesis of RNA, while deoxyribonucleosides are responsible for the synthesis of DNA there. So basically whenever ribonucleotide reductase is getting inactivated, the level of ribonucleotide gets accumulated, while the level of gets increased because of its accumulation, while the level of deoxyribonucleotide gets decreased because it is not getting produced from the ribonucleotide there, right? So this is basically this hydroxyurea actually binds to this ribonucleotide reductase, leading to this particular accumulation of ribonucleotide and reduction of deoxyribonucleotide or DNTPs there. So what hydroxyurea does is that it also has a property of undergoing suicidal death of cells, or what is called a suicidal apoptosis. So anyone of you tell me that there is a particular component to which hydroxyurea needs so that it can undergo this suicidal apoptosis? Can anyone of you tell me which component binds with hydroxyurea so that the cell can get arrested at the G1 phase at the start checkpoint, and furthermore it can undergo suicidal apoptosis? What was that component? Basically we have discussed in the earlier one of the assignment sections. So I think some of you might have remembering the word caffeine, right? Caffeine as well as caffeine alone cannot undergo suicidal apoptosis, but hydroxyurea binds with this ribonucleotide reductase, inhibit its function. But hydroxyurea along with caffeine, what it does is that it actually undergoes the cell, tells the cell to undergo suicidal apoptosis. So what happens is that the cell will get stuck at the start checkpoint in the late G1 phase, and afterwards, because of the presence of both of these components, the cells start to undergo suicidal apoptosis that leads to cell death and all. So that's also a particular functionality of the hydroxyurea when it combines with caffeine. Along both of these components cannot do that basically. So that's the basic answer. Hydroxyurea is an inhibitor of ribonucleotide reductase. So anyone has any doubt regarding this? This we have discussed in the previous week section, assignment sections and everything. Please go through that also. Yes, basically what hydroxyurea does is that hydroxyurea has a tendency to bind to this ribonucleotide reductase. So ribonucleotide, if you see the functionality of ribonucleotide, what it does is that it helps in the conversion of ribonucleotide to deoxyribonucleotide or DNTPs. Ribonucleotides are essential for the formation of RNA, while DNTPs are essential for the formation of DNA. So whenever ribonucleotide is getting inhibited in the presence of hydroxyurea, this level of ribonucleotides get increased, NTP get increased, while the level of DNTP gets decreased because it is getting inhibited there. There is no longer production of DNTP, while ribonucleotide is getting used up, is not getting used up there. So that's the functionality of hydroxyurea. It actually inhibitor of ribonucleotide reductase. So however, whenever hydroxyurea is combined with a specific compound called of caffeine you know that is present in tea and everything, right? Coffee, it is present there. So caffeine alone cannot lead to cell death, but whenever caffeine or hydroxyurea combines with each other, whenever both of the hydroxyurea or caffeine are present with or added to the cell in the or present the organism, what it does is that it is getting absorbed by the cell, so it promotes the cell to undergo a process called suicidal apoptosis. So what suicidal apoptosis does is that the cell gets stuck at the start checkpoint at the G1 phase there, right? So start checkpoint is present at the late G1 phase. It in order to cross the start checkpoint, whenever the start checkpoint is getting crossed, the G1 phase is getting transition into S phase there, right? So whenever caffeine is provided along with hydroxyurea, the cell gets arrested at the G1 phase and it undergoes a particular process called as suicidal apoptosis. So that means it undergoes cell death there. So hydroxyurea alone cannot do this, caffeine cannot also alone do this, but whenever both of them are combined together, this there is a particular function that is carried out by both of them, which is called a suicidal apoptosis that leads to cell death. So start checkpoint is not getting crossed there, it leads to cell death there. So in the previous week section, I think we have discussed there the possible answer of how this mechanism happen. I think I have discussed in that case. So please go through that also. So I hope I am clear in this case. So should I repeat that again? Okay, so I think it's clear. So I will just move on to the next question. Okay, coming on to the next question. Which of these proteins associated with helicase and helps it to load it at the origin of replication? CDC7, CDC5, CDT1 or CDT2, which may be the answer. Which of these protein helps to load the helicase into the replication origin? Okay, so I think I will just give you the answer. So answer to this question is CDT1, that is option number 3. So basically this is the diagram there. You can see that I will just open up the pointer there. you can see that this is the origin of replication that is present here, which is already occupied by these two complexes, that is origin of replication complex as well as CDC6. So whenever the MCM helicase, whenever the replication need to start, what happens is that I think at the early S phase and everything, like MCM helicase need to be loaded out. So what happens is that this CDT along with this CDC6 or CDT actually binds to this MCM helicase there and actually recruits the particular MCM helicase to both the sides of origin of replication complex. So this diagram is quite different basically. So one MCM helicase will be present at one end and the other MCM helicase will be present at the other end. So that leads to the fact that two MCM helicases are loaded into origin of replication complex. One moves in the opposite direction in the left direction there, while the other one moves in the right direction, so that the replication bubble is formed there, right? So that's a function of helicase there. So this CDT1 actually helps in the loading of helicase into MCM complex, and then CDC6 actually helps in the release of the CDT1 that is bound to MCM1, so that it becomes free there. You can see that the release of CDT1 happening there. So one MCM helicase bound to one side of origin of replication complex, while the other one binds to other side. So both moves in the unidirectional manner in opposite direction, so that the replication bubble is initiated starting from the origin of replication. So yeah, so there are 2 to 7 helicases there. About I think 8 helicase complexes are present here. The 8 helicase subunits are present here. So that's why it's called as MCM 2 to 7. So and these two together with this MCM helicase as well as origin of replication complex is called as a pre-replicative complex. So I hope I am clear. So anyone has any doubt? CDT1 helps in loading MCM helicase into the origin of replication complex or origin of the replication. Okay, so I think it's clear, so I will just move on to the next question. Coming on to the next question. GTP hydrolysis take place in which part of the microtubule filament? Alpha tubulin, this is a tubulin basically, so I forgot to write that. Alpha tubulin, beta tubulin, gamma tubulin, or all of the above. In which in part of the microtubule GTP hydrolysis take place? It is required for polymerization and everything, GTP hydrolysis. But to one of the tubulin subunit it's actually binding. Two of the tubulin subunit is actually binding, but only in one of the tubulin subunit GTP hydrolysis take place. Out of which of the tubulin subunit might be carrying out this function? Yeah, I'm getting option number 2. Okay, so basically you are right, beta subunit, right? So basically alpha and beta both bind to the GTP subunit, while gamma is actually acting as the microtubule polymerization tubulin, right? Polymerization subunit actually polarizes the microtubule so that this microtubule assembly can take place. While afterwards alpha and beta binds to it, while alpha and beta both have the capability to bind to GTP, but alpha binds more tightly to GTP as compared to beta, which binds it too loosely. However, beta subunit is the one that carries out this GTP hydrolysis, so that polymerization takes place. So GTP hydrolysis microtubule primarily occurs in the presence of this beta tubulin subunit, and it has an exchangeable GTP binding site, which is also called as an E site, where GTP is hydrolyzed to GDP during or shortly after polymerization. The alpha tubulin also has a non-exchangeable GTP site which binds tightly to GTP. However, GTP hydrolysis cannot take place in this case. So that's the basic function of both of these tubulin subunits that are present in a microtubule. So I hope I am clear. So anyone has any doubt regarding this? Okay, so I think it's clear, right? So I will just proceed on to the next question. Okay, this we have discussed before. Which of these amino acid is getting phosphorylated to make the CDC28 active? Threonine 40, threonine 161, tyrosine 15, both tyrosine 15 and threonine 161. Which may be the answer? CDC28 is also called as CDK1. It binds to cyclin B. In case of yeast, I think Saccharomyces cerevisiae, it is called as CDC28. Which is the activating phosphorylation that is happening here. To which amino acid the phosphorylation is happening, so that CDC28 or that CDK kinase is getting activated. Okay, I am getting option number 4, both tyrosine 15 and threonine 161. Okay, so the answer to this question is threonine 161, right? So basically, I will repeat this diagram once again. I think this is an important diagram there. So in case of CDC2, in case of Saccharomyces pombe, in case Saccharomyces cerevisiae, it is called as CDC28. Please check that out because it might be opposite also. I don't also recall the correct nomenclature there. So in case of CDC2 or CDC28, there is threonine 161, tyrosine 50, as well as threonine 14 there, right? So this kinase, CAK kinase, which is a kinase, cyclin activating kinase, and V1 kinase actually helps in loading of cyclin into the CDC2 by phosphorylating these three. So these three amino acids are getting simultaneously phosphorylated by these two kinases there. So two of them are actually inhibitory in nature, this tyrosine 15 as well as threonine 40. So we have discussed that here, right? Option 1 and 3. While threonine 161 is called as the activating amino acid there. So CDC25, which is a phosphate, actually removes this inactivating phosphorylation there. That is, it removes the phosphate groups from this tyrosine 15 and threonine 14, rendering it free or dephosphorylated, while the threonine 161 is still dephosphorylated in this case. So the activating amino acid is getting phosphorylated there. So whenever this happens, it is becoming active there. This cyclin B, CDC2 or CDC28 or CDK1 complex is activated there. So the mitosis is getting regulated at this phase there because the active cyclin B complex there, cyclin B or CDK complex there. So whenever this phosphorylation is getting removed by this, what is a anaphase promoting complex and all, this cyclin is getting degraded by proteasomes and all, which actually transition from the metaphase to anaphase and finally into a G1 phase. So out of this amino acid is getting phosphorylated, threonine 161 is getting phosphorylated, which renders the CDC28 active. So I hope it is clear. So anyone is in doubt? Okay, so I think I will just move on to the next question. Coming on to the next question. What is the number of amino acids that are present in a polyubiquitin tag? 76 amino acids, 72, 82 or 78? The number of amino acids that are present in a polyubiquitin tag, it is involved in polyubiquitination and a proteasomal degradation, right? So how many amino acids are present in this polyubiquitin tag? Oh, sorry, like how many ubiquitin is present in this polyubiquitin tag? I will just change that. Okay, so the answer to this question is 76. So basically the polyubiquitin tag is getting attached to what you say, the particular N-terminal end of the amino acid that protein that needs to be degraded there, right? So there is a particular sequence that is present there, which is called as a destruction box sequence there. So to which the ubiquitin ligase is getting recognized there. Ubiquitin ligase recognizes this particular motif there and binds to it and adds this polyubiquitin tag there. So about 76 polyubiquitin molecules are getting added there. That's why it's called as a polyubiquitin there. It's similar to what is called as a polymer there. There are about 76 in number. So whenever this polyubiquitin is getting added there, it is recognized by these proteasomes. And furthermore, whenever it is recognized, it is going into a proteasome, and the protein that is attached to this polyubiquitinate tag is getting degraded out and released into individual peptides or amino acids there. So there are 76 ubiquitin that is present in this particular ubiquitin tag there. So I hope it is clear. I will move on to the next question. Coming on to the next question. In plant cell, fusion of which organelle lead to formation of a cell plate? Endoplasmic reticulum, vacuoles, Golgi, or mitochondria? Fusion of which organelle leads to a formation of cell plate in case of plants? Cell plate is required for cytokinesis, right? It is formed by a fusion of one of these organelles. Okay, so the answer to this question is Golgi or Golgi bodies. So this is the basic diagram. So you can see that at the telophase stage, the cell plate needs to be formed there. So what happens is that the Golgi vesicles that are found in this cell is accumulated at this equatorial plane there, and they fuse together to form this cell plate. So this cell plate mainly consists of this, what you say, the middle lamellae and everything. So you can see that the Golgi bodies fuse together, they get thickened and form this cell plate at the equatorial position there. Afterwards, it gets connected to the primary cell wall, and the middle lamellae is getting degraded and the primary cell wall is getting connected. So you can see that there is two different cells that are present within a single mother cell there, right? Afterwards, the cytokinesis takes place, so that cytokinesis happens, so that you can see that the two different cells are present there, which are joined together by a cell wall there. So this is how a cell plate form. It is actually formed by the fusion of cell plate at the equatorial position, leading to the formation of a cell wall in between, dividing the two cells there. Case of animals, there is like I think cleavage furrow and everything that is present there, right? So that leads to the formation of that leads to cytokinesis that is happening there. So I hope I am clear. So anyone has any doubt regarding this? Okay, so I think I am clear. I will just move on to the next question. Okay, coming on to the next question. Activation of DNA helicase take place at which phase of the cell cycle? S phase, late M phase, early G1 phase, or G2 phase? Activation of DNA helicases. Yes, I am getting option number 1. Anyone else? Okay, so the answer, you are right. Basically, answer to this question is S phase, that is option number 1. So basically, in case of like the loading actually take place in the like late G1 phase or in the early S phase there, right? I think, yeah, in the late G1 phase. So the helicases are getting loaded into the origin of replication complex, near to origin of replication complex at the DNA origin. So whenever it is loaded, afterwards whenever it is transitioning into the S phase, whenever the start checkpoint is getting passed on there, G1 to S phase translation time space. So afterwards the helicases are getting active there. So in the late G1 phase, the helicases might be present at the origin of replication there, but it is not active. The cell need to transition into S phase, so that DNA replication need to begin. Only then the MCM helicase is getting activated there, so that the replication, after activation, the replication bubble can form, followed by initiation of DNA replication there, right? So that's how the process goes. In the late G1 phase, MCM helicase is getting loaded there. In the S phase, it is getting activated. Once activated, replication bubble form and DNA replication is getting initiated there. And because of the transition from G1 to S phase, because after passing the start checkpoint and all. So anyone has any doubt regarding this? Okay, so I think I'm clear. I will move on to the next question. Two ubiquitin in a polyubiquitin tag can be attached to each other via which of these amino acid interaction? Lysine 48 or lysine 53, lysine 48 or lysine 46, lysine 63 or lysine 53, lysine 48 or lysine 63. So, a polyubiquitin chain is interacted by interaction actually take place because of the bonding that is formed between two ubiquitin between two amino acid residues. So that's how a polyubiquitin chain is formed. Which of these amino acids interact with each other to form that polyubiquitin chain within the ubiquitin itself? Yes, I am getting option number 4. Anyone else? Okay, so you are right. The option number 4 is the right answer, lysine 48 or lysine 63. So if you consider two ubiquitin chains there, lysine 48 of one ubiquitin can interact with lysine 48 of other ubiquitin chain, or it can interact with lysine 48 of one ubiquitin can interact with lysine 63, vice versa. Or lysine 63 of one ubiquitin can interact with the lysine 63 of another ubiquitin, so that the whole polyubiquitin chain is getting formed there. So basically that's how a ubiquitin is getting attached to each other to form that polymer of ubiquitin called as polyubiquitin tag there, which is getting attached to certain protein which need to be degraded out. So I think this is clear. So anyone has any doubt? Okay, so I think it's clear, right? So I will just move on to the next question then. Coming on to the next question. Pheromones like alpha factor produced by yeast activates which pathway? Beta catenin signaling pathway, Hedgehog signaling pathway, MAP kinase cascade pathway, or no signaling pathway? Which of this might be the answer? So this is involved in meeting switching in case of yeast and Saccharomyces cerevisiae, right? So there's a particular pathway that is activated by this particular pheromones there. Out of which of this might be the answer? G-coupled protein receptor pathway, similar to this, several are there, right? Out of this, which might be the answer? So the answer to this question is MAP kinase pathway, right? So what happens is that whenever this pheromone, there is a alpha strain, there are different strains of yeast, right? Consider that this is an alpha strain of yeast. So alpha strain of yeast releases this alpha pheromone whenever it comes in contact with another alpha strain of yeast. So similar strains of yeast comes together, then one of the strain will, or the both of the strains start to release this kind of pheromones and all. So whenever alpha factor pheromone is released from one yeast, it binds to the receptor, that is the GPCR, G protein coupled receptor, which consists of several transmembrane helixes are seen here. To this particular receptor there, the pheromones actually bind to the other yeast strain. So it is getting detected there. Whenever it detects that it is a particular pheromone that is released by an identical strain of yeast, that is an alpha strain of yeast, this actually pathway gets activated. What happens is that the alpha subunit of this GPCR is actually bound to this gamma as well as beta subunit there. So whenever it is pheromone is getting detected or found to be identical, this G beta as well as gamma subunit is getting released from there. So this G alpha is getting bound to this receptor complex itself, while the G beta as well as G gamma is getting released there. So this in turn activates some additional downstream pathways, including binding to this particular protein complex there, that is T5, T11, T7. You don't need to go detail into it. I was just showing you the pathway there. So whenever it becomes active, it actually activates other downstream proteins like FAR1, which inhibits the G1 arrest. So G1 arrest is actually essential for preventing the transition into S phase. So whenever the cell is getting arrested in the G1 phase, it has a tendency to bind or fuse with the other, say, identical strain of yeast, whichever is getting activated here similarly, and then fission can happen there, right? So these two cells join together, haploid cells join together to form a diploid cell. Diploid cell then undergo meiosis to form haploid spores, and then haploid 4 germinated into another haploid strains of yeast there, right? Alpha strains of yeast there. So that's how this function actually take place. So FAR1 is getting activated there. This FAR1 inhibits the transition from G1 phase to S phase, promoting this G1 arrest there, and also it also inhibits the formation of this, like what you say, one TIC2 protein, which in turn inhibits this case. So whenever this protein is getting activated, inhibited there, this actually getting activated, T12 protein is getting activated there. So this transcribes additional transcription factors or proteins that leads to this G1 arrest and all. So this is how this pathway actually works. You don't need to go detail into it. I am just showing the mechanism of how it is working. So it activates this MAP kinase cascade to GPCR or G protein coupled receptor complex, and that's how this mating switching happens by like that homologous recombination that we have discussed before, right? By MAT1 locus and all. These are actually interconnected with each other by this pathway. So yeah, so MAP kinase pathway is activated by pheromones like alpha factor produced by the yeast by binding to GPCR or G protein coupled receptors. So I hope this is clear. So anyone has any doubt? Okay, so I think it's clear, right? So I will just move on to the next question. Coming on to the next question. What is the function of a T loop in human CDK2 or cycling dependent kinase 2? Did anyone ask me down? Okay, basically, sorry for that. So basically, these are other pathways that I just gave as an option. So there are several signaling pathways that are taking place in vertebrates and all. Node signaling pathway, as you know, is involved in apoptosis and everything, right? In order to mediate the extrinsic apoptosis pathway and all. Node signaling pathway is also called as death signaling pathway, which lead to activation of extrinsic pathway. Beta catenin is also involved in like maintaining the cell proliferation and cell division. Like several beta catenin proteins are produced there, which are involved in cell division pathway. So Hedgehog pathway also has a similar activity, but it is something that is related to cancer. It has several receptor pathways that are involved in pathway. So these are basically highly detailed in this case. So I don't also recall the correct pathway that is actually coming out of this. So this might be happening in yeast in some other pathway also, but most of this beta catenin, Hedgehog and signaling pathway are happening in case of like vertebrates and all, except for no signaling pathway which is involved in apoptosis. So I think there is no need. Basically, I just give you as an option in this case. So if you are interested, you can just go through some of the important books that actually gives this important cell signaling pathway widely in a descriptive manner. So at this point, I don't recall the correct functioning of all this pathway. So I hope I am clear. Okay, thank you. So I will just move on to the next question. We have asked this before, right? What is the function of T loop in human CDK2? It acts as the active site of the kinase. It blocks the access to active site. It binds to ATP near the active site, or none of the above. Which may be the answer. what is the function of T loop in human CDK. Okay, so the answer to this question is that it blocks the access to active site. So basically this is a structure of human CDK too. So you can see that these are I think parallel beta sheets, anti-parallel beta sheets, and these are alpha helices that are present here. You can see the random coils that are also present here, right? And you can also see the ATP molecule that is present here. So p star helix is actually found in this alpha helical loop that is found here. While you can see that there is a T loop we are discussing here, right? So the T loop actually exists at this particular region there. So what happens is that this particular region actually has the tendency to block the active site. It is flexible in nature. So whenever this particular CDK is active, the T loop is actually flexible and in an open manner. So ATP can go into the active site and it can actually help in carrying out the phosphorylation activity, right? So it is a kinase, cycling cell-dependent kinase handle. Whenever the ATP is present, it can actually carries out this kinase activity. But the T loop needs to be open in this case because of the flexible nature of this T loop. It is opening the active site. ATP can go in and it can carry out the phosphorylation activity. So that's the one of the active site of CDK2. While p star helix is close to the active site in the kinase, and ATP binding site actually take place between this. We can see that the parallel beta sheets and all, right, near the active site and all, in the active site. So the T loop functions as the the flexible gatekeeper of the particular active site of the complex, right? It needs to it can be either be open or in a closed conformation. Whenever it is open, ATP can go in and activate a function. Whenever it is closed, ATP cannot enter into active site and it renders inactive. So I hope I am clear. So anyone has any doubt? Okay, so I think it's quite clear, right? So I will just move on to the next question. Coming on to the next question. Which of these site in a proteasome unfolds protein so the cell can enter into it? Unfold protein so the protein can enter into it. Regulatory subunit, AAA ATPase module, core particle, or none of the above. Which of these unfold the protein so that it can enter into the proteasome out of all these subunits in a proteasome? Anyone? It's an easy answer, right? So the answer to this question is AAA plus ATPase module. So this is the structure of a proteasome. The 26 S means the Swedberg units, right? By ultracentrifugation or sedimentation velocity that is characterized by this protein, the molecular weight is determined as 26x. So it consists of this 20s catalytic core complex or 19s regulatory complex. So the regulatory complex consists of this base subcomplex and lid subcomplex also. So considering the lid subcomplex, the lid subcomplex is also called as the regulatory subunit. So what does it do is that it detects this protein consists of this polyubiquitin tag there. So this is the polyubiquitin that is attached to the protein, right? So it is getting detected by this regulatory subunit. Afterwards, when it is getting detected, the protein actually is getting unfolded by this AAA ATPase module that is present within the base subcomplex there. This is the base subcomplex there. So this AAA ATPase module is present there, and whenever this regulatory subunit detects this particular polyubiquitinated protein, it unfolds the protein so that it can enter into the core complex there. So the core complex actually consists of act as a protease there. So the protein needs to be degraded out there, right? So that's the function of proteasome. So the regulatory subunit detects this polyubiquinated protein. The AAA ATPase unfolds this protein, and it allows the entry of this unfolded protein into a core particle which acts as a protease there, degrades the protein out into specific peptides or amino acids there. You can see the degraded protein coming out of the other side of the proteasome. That's how it functions. The regulatory subunits detects this polyubiquinate protein. AAA ATPase unfolds this protein and then inserts itself into a core particle, where the core particle degrades the protein because of its protease activity, and the degraded protein comes out of the proteasome to the other side. So this is how the proteasome actually functions in the case. In the basic language, it is how the proteasome functions. So I hope I am clear. So anyone has any doubt, or should I repeat it once more? Okay, so I think I am clear. I will move on to the next question then. Coming on to the next complex. In which of the following phase of the cell cycle does licensing of pre-replicating complex take place? S phase, late M phase, early G1, both 2 and 3. Licensing of pre-replicating complex take place at which phase of the cell cycle? Yeah, so basically you're right. The answer to this question is both 2 and 3, that is late M phase as well as early G1 phase, right? So licensing is a term that is used to uh, basically what it means that the origin of replication is getting incorporated by this origin of replication complex and followed by this MCM helicases there, right? So it is not initiating the replication process, but all the components that are needed for starting the replication is getting incorporated into the replication origin there. That is the MCM helicases, that is MCM 2 to 7 helicases, as well along the origin of replication, which leads to formation of pre-replicating complexes there. So whenever this pre-replicating complex is present at that stage, that is called as a licensed stage of the cell cycle, which happens at the late M phase as well as early G1 phase. So I hope I am clear. Anyone has any doubt? Okay, so I think it's clear, right? So I will move on to the next question. Sorry for that, I need to, yeah. So the answer is quite clear. So I will just read out this because I haven't properly edited this slide. So anaphase promoting complex is activated by binding to which of these following? CDC20, CDC15, CDC1 or CDC3. So from that answer you can get that the answer is CDC20 there. So basically anaphase promoting complex needs to be active at the whenever a metaphase to anaphase transition need to take place, right? That is a late M phase there. So what happens is that this CDC20 gets bound to this phosphorylated, the anaphase promoting complex is getting phosphorylated by cyclin B, CDK1, and after when it is phosphorylated, what happens is that this CDC20 binds to it and it then activates the anaphase promoting complex or cyclosome complex there. So it's actually acting as a regulatory cofactor there. So this interaction enables the ABC anaphase promoting complex to ubiquitinate key cycle regulators cyclin, securin as well as mitotic cyclin, including cyclin B, which is needed for maintaining the M phase and triggering their degradation by proteasomal degradation mechanism and followed by the driving the progression through metaphase to anaphase stage, which helps in the separation of this sister chromatids there. It binds to the phosphorylated APC complex as we had this in the early mitosis, activating E3 ubiquitin ligase activity to degrade the substrate containing a destruction box box motif, right? So this D box motif is getting polyubiquitinated there. So afterwards, because of polyubiquitination, it is recognized by this the proteasome and getting degraded out. So this is the active function. So these are all some extra points. You can just go through that. I will give you the slides there. So based on the this particular option, does anyone has it out? I'm sorry that I didn't edit this slide, so the answer already came up when the question was asked. So, however, anyone has any doubt? Okay, so I think it's clear, right? So I'll move on to the next question. Coming on to the next question. Which of the following statement is incorrect? S phase cell induces premature DNA synthesis in G1 phase. S phase cell induces DNA synthesis in G2 phase. Cytoplasm from M phase cell promotes entry of oocyte into M phase. Cytoplasm from interphase cell does not promote the entry of oocyte into M phase. Which may be the answer? Which of this following statement is incorrect? Okay, so the answer to this question is sorry. Okay, this also I did. Sorry for that. The answer to this question is S phase cell induces DNA synthesis in G2 phase. So this is the incorrect statement. If you see the first phase, the S phase cell induces premature DNA synthesis in G1 phase, because whenever the S phase cell is cytoplasm from the S phase is introduced into G1 phase, the S phase is already in a pre-replicative phase, right? It undergoing replication at all. So the components that are present in the cytoplasm is introduced into G1 phase. So it transitions from the start checkpoint and enter to G1 phase so that the DNA synthesis can take place. So this option is correct. In case of option number 2, the S phase cell induces DNA synthesis in G2 phase. That is incorrect, because the cell division is always in a unidirectional manner, right? It's in a unidirectional cyclical manner. It can go from G1 phase to S phase to G2 phase, M phase, and then into G1 phase, and vice versa. So, even if you introduce a cytoplasm from S phase into a G2 phase, what happens is that it cannot induce a DNA synthesis, because G2 phase comes after S phase and it cannot revert back into that previous phase that it was gone through. So that statement is incorrect. So cytoplasm from N phase cell can promote entry of oocyte into S phase, because the oocyte is arrested in the G1 phase, I suppose. So whenever M phase cytoplasm is introduced there, it can pass through the S phase and then into the M phase, so that the mitotic can the cell arrest can be ignored and mitosis phase can be started there, because M phase consists of this maturation promoting factor and everything that comes in the first speaker's presentation, right? So that promotes the entry into cell from the cell arrest stage into M phase stage. Cytoplasm from interphase cell do not promote the entry into oocyte into M phase, because the oocyte is already in the interphase stage. So even if we are introducing the cell cytoplasm into oocyte from the interphase cell, it cannot promote M phase entry. So yeah, so the answer is incorrect statement is S phase cell induces DNA synthesis in G2 phase. No, it cannot induce DNA synthesis in G2 phase. So I will just correct this out. Sorry for that. So I hope it is clear. So I will just move on to the next question. Okay. Coming on to the next question. Start checkpoint regulates which transition in the cell cycle phase? Which might be the answer? G2 to M1 phase, G1 to S phase, S to G2, or M to G1 phase? Okay, so this we have discussed before, right? So the answer to this question is option number B, G1 to S phase. So I will move on to this is the phase. Basically, you can see that the G1 phase, you can see that this is a checkpoint, which is also called a restriction point or start checkpoint there. So it need to pass through a checkpoint so that it can transition from G1 to S phase. Okay, so I will just give you the final question. So final question is that which of this following statement is incorrect? Retinoblastoma is incorporated with E2F and it inhibits its function. Dephosphorylation of retinoblastoma allows its release from the E2F transcription factor. It helps in the transition from G1 to S phase, this retinoblastoma E2F transcription factor interaction. E2F acts as a transcription factor and promotes activation of S phase genes. Which might be the answer? Which of the following statement is incorrect? Okay, so the answer to this question is incorrect statement is dephosphorylation of retinoblastoma allows its release from ETOs. So that's basically phosphorylation of retinoblastoma protein allows its release from ETOs. That should have been the correct answer there. So whenever retinoblastoma is bound to this ETOs transcription factor, the transcription activity of this transcription regulator activity of ETOs is inactivated there. However, when the cyclin CDK proteins are getting activated there, whenever G1 to S phase transition needs to take place, certain CDKs actually phosphorylate this retinoblastoma. You can see that the site is getting changed there. So this results in the release of ETOs transcription factor there. So this ETOs transcription factor actually binds to transcription factor certain promoter regions in the genes that are required for S phase cyclin generation and all, including S phase cyclin generation and all. This ETOs binds to its promoter and leads to formation of to a production or translation of S-phase cyclin and all. So that leads to a crossing of the start checkpoint, that is from G1 to S-phase checkpoint. So that's the basic function of this particular E2 transcription factor. So the answer is it should have, this is incorrect option. Phosphorylation of retinoblastoma allows its release from E2. That's the right answer. So anyone has any doubt? Okay, so I think it's quite clear, right? So thank you for everyone for attending this class. So based on the cumulative of this class, does anyone has any doubt from what all we have took today? Okay, so I think there are no doubts, right? So I have attached each and every presentation and YouTube presentation recording to the NPTEL link, so you can go through it and prepare well for the exam, and best of luck. This is the last section. Thank you.